Hi guys, welcome to our exploration into the world of anti-cancer drugs. Cancer, a disease that knows no boundaries, affecting millions worldwide. The quest for its effective treatments has been an ongoing battle, pushing the boundaries of medicine and science. Let's get some overview about cancer. It is a disease resulting from uncontrolled growth of abnormal cells within the body. In cancer, there is a formation of tumor, which are the groups of abnormal cells that form lumps in the body. Genetic changes can also lead to cancer if they alter the way your cells grow and spread. Here you can see how a tumor is formed. The genetic change in normal cell leads to cancer cell, which upon doubling forms malignant tumor. The tumor impacts the normal body function by causing some changes in the body or may block the channels in the body. It can stop particular body systems too. Before going towards the treatment or anti-cancer drugs, it is necessary to know that early diagnosis is really important to have the best chance for successful treatment. If it is delayed, there are less chances of survival. Any drug that is effective in the treatment of cancerous disease is an anti-cancer drug, and these are used in various cancer treatments, which includes chemotherapy, targeted therapy, immunotherapy, and hormone therapy. Chemotherapy is specifically associated with the use of anti-cancer drugs to kill the cancer cells. Targeted therapy uses drugs to target specific genes and proteins that help cancer cells survive and grow. Immunotherapy is a treatment that helps immune system to fight cancer. And what is hormone therapy? Hormone therapy is a type of cancer treatment that removes, adds, or blocks specific hormones to the body. Anti-cancer drugs may be used before or after the surgery or radiotherapy, depends on various factors. Some other cancer treatments include biomarker testing, which is a way to look for genes, proteins, and other substances that can provide information about cancer. Hyperthermia is a treatment in which a body tissue is heated to help damage and kill the cancer cells. Photodynamic therapy, as indicated from the name photo, involves light source and light sensitive medicine to kill the cancer cell. Stem cell transplant is a medical procedure in which healthy stem cells are transplanted into the bone marrow or blood.
We have two types of anti-cancer drugs: cell cycle specific drugs and cell cycle non-specific drugs. Cell cycle specific drugs are those which exert their action on cells in specific phases of cell cycle. Example: 5-fluorouracil and bleomycin. While cell cycle non-specific drugs are those which exert their action on cells whether in any phase of cell cycle or G0 phase. Anti-cancer drugs are classified as alkylating agents, which add alkyl groups to DNA, damaging the DNA of cancer cells. As cancer cells are more sensitive to DNA damage than healthy cells, their DNA damage leads to cell destruction, while these drugs also damage healthy cells. Next, we have antimetabolites. They interfere with the DNA synthesis, resulting in the damage of DNA. Next, cytotoxic drugs, which inhibit cell division and ultimately leads to cell death. Plant derivatives drugs, which are the drugs derived from plants and are toxic to cancer cells. The hormone drugs target hormonal functions responsible for the production of cancer cells, while the protein kinase monoclonal antibodies and miscellaneous agents are also responsible for the death of cancer cells. These are some examples of classes of anti-cancer drugs. We will talk about the mechanism of anti-cancer drugs and what causes the cells to become cancerous or tumor cells. We all know that whenever a cell urges to divide, it enters the cell cycle. The cycle which divides a cell into two daughter cells. Cell cycle contains certain phases like G1, S and M phase. Some cells also enter G0 phase for a short period while others enter it permanently. G1 and S phases are the preparatory phase, while M phase is the dividing phase, which divides the cellular contents into two daughter cells. Once a cell completed its division, there happens two possibilities for it. Either it exits the cell cycle and becomes a dormant cell, or it again enters the cell cycle to divide again. To ensure proper division of cell, there are certain checkpoints at different phases of cycle. These are at G1 phase, S phase, and M phase. In case of genetic mutations in some cells, they skip the checkpoints or pass these checkpoints with a great speed. In this way, they continue to divide abnormally and become tumor cells. To treat them, we use anti-cancer drugs. Let's start with the action of drugs in G1 phase. In this phase, cell synthesizes necessary proteins for DNA replication and cell division. The main building block amino acid for these proteins is asparagine. In the case of healthy cells, asparagine is synthesized by the help of enzyme asparagine synthetase. While the tumor cells, such as leukemic cells, lack this enzyme and depend on exogenous supply of asparagine for their survival. At this point, the game of anti-cancer drugs starts. The drug which is applied in this case is asparaginase, which can convert asparagine into aspartic acid and ammonia, leading to the depletion of asparagine, inhibition of protein synthesis, and ultimately apoptosis or cell death in leukemic cell population. In some cases, asparaginase may cause allergic reactions in patients. For this purpose, scientists developed advanced version of asparaginase called pegaspargase, which is less likely to allergic reactions, has longer duration in the body, and can be given less frequently.